All right, guys. We are looking at passive transport. And again, passive transport doesn't use any energy. So things are going to move from an area of high concentration to an area of low concentration. But you need to remember that molecules take and they move randomly. So in lecture, I talked about the scent particles that are leaving my son's rear end and how they diffuse from an area of high concentration to low concentration until they reach this equilibrium, and then the net movement is going to be static. So like there isn't going to be an overall noticeable net change in concentration of particles. This is a video that you can watch on Diffusion that takes and explains things a little bit better, hopefully, for you. If you need to watch that, go ahead. And this is talking about, this picture right here is talking about how the diffusion across a substance of a substance across a biological membrane is a passive transport because there's no energy expended by the cell, okay? And the concentration gradient represents the potential energy that actually drives that diffusion, right? The rate of diffusion depends on the membrane permeability. So the permeability of what is setting up that actual gradient, okay, to that specific substance. So depends on whether things can actually move through that gradient itself. And substances are going to diffuse down their concentration gradient, and they're going to move in relation only to their own concentration. So when I talked in lecture, I equated this to the fact that my son, when he passes gas, I smell the smell when I am driving my car. But then my daughter, who wants to be like my son, she also passes gas, and I can smell that smell also, okay? Because those two scent particles are going to diffuse independently of one another. Osmosis is just the movement of water for a concentration gradient. So it's the diffusion of free water. So water molecules not clustered around another substance across that selectively permeable membrane. So the water is going to go from where it is in high concentration to where it is in lower concentration, okay? So I use to remember which way water is going to move. I always tell myself salt sucks. So wherever the solute, in this case salt, okay, well in this picture it's sucrose, okay, wherever it is in higher concentration, it's going to suck the water towards it. So the water is going to move towards where the solute is in higher concentration, because with the solute being in higher concentration, the water is in lower concentration. So again, the net movement of those water molecules is going to go where there is a higher concentration of the solute in solution. This is taking and showing that we have this semi-permeable membrane at the bottom of this tube. So it's selectively permeable. It is allowing water through, but it does not allow the actual sugar molecule through. And since we're here on the left, you have a lower concentration of the solute, so a lower concentration of the sugar. And on the right, you have a higher concentration of the solute or a higher concentration of the sugar. So water is going to move in the direction of where you have that higher concentration in order to dilute that concentration, because the molecules, the particles can't move. So the water has to move instead. So again, this takes and shows that a little bit more closely, that water molecules can pass through the pores, but sugar molecules cannot. And then the water molecules cluster around the sugar molecules. And so this side has more solute molecules on the right-hand side and fewer free water molecules. And on the left-hand side, there's fewer solute molecules and more free water molecules. And so the water is going to move from where it's in higher concentration of free water molecules to where to where there's less free water molecules. And so osmosis, again, is the movement of water. And again, this is a passive process, meaning it doesn't use energy. This is a video that talks about it. And then we have tonicity. Tonicity is the ability of the surrounding solution to cause a cell to gain or lose water. We looked at this in lab. So in a hypertonic solution, you put a cell in a hypertonic solution, water is going to take and move out of the cell because the water is in higher concentration inside the cell, and it's going to move outside the cell to where there's more of the solute. So the salt sucks, and we're taking the water is flowing out of the cell, and that results in plasmolysis, which causes the cell to shrink. In a plant cell, you have that plant cell wall that it stays stable. So it is a stable structure and it's not going to shrink. But the cell membrane will pull away from the cell wall, okay, and kind of collapse on itself, similar to what happens in an animal cell. But again, that cell wall is still going to be visible at the same size. If we take and we put it into a cell into a hypotonic solution, okay, meaning that there's a lower concentration of solutes outside the cell than inside the cell, okay, so hypo meaning lower, so lower concentration outside the cell than inside the cell, then water is going to flow into the cell. And that can cause cytolysis and actually like cell lysis, like it could, a cell could rupture, okay? And then in an isotonic solution, that's where you have an equal number of solutes inside and outside, equal number amount of water inside and outside of the cell. That is the overall net movement. There isn't any. So water is equally moving in and out of the cell, okay? And that's where things are generally a little bit happier because the cell doesn't have to work on trying to get water out, okay? And that would be an energetic process if it's going against a concentration gradient. So this takes and shows you an animal cell versus a plant cell. And again, look at how that plant cell wall stays rigid. And so you can take and the cell membrane will either pull away from the cell wall when it's plasmolized, or it will actually take and push up if it's turgid, which is a normal state generally for a plant. And then up above that is a red blood cell in animal cells. And when it was placed into a hypotonic solution, it was lysed, meaning it exploded. And then if you place it in a hypertonic solution, it shrinks. So this causes problems for animals that are aquatic. Because they can cause these cells can't tolerate excessive water loss or uptake because they don't want to shrivel and they don't want to explode, okay? Organisms that live in these aquatic environments, they actually have to do osmoregulation, which they're controlling a solute concentration and water balance, okay? And so they have to have a method in order to deal with this. So in a paramecium, the paramecium has a contractile vacuole, okay? And that contractile vacuole is actually pumping excess water out of the cell. And so that the paramecium is using energy to get water outside of the cell so that it doesn't like swell and burst. And so that itself is something that is beneficial and something that the paramecium is worth, it sees value in spending energy on. There's a video of how a paramecium vacuole works, and you might actually take and enjoy watching that. And here's a video of Eladia, and we saw this in lab also, where water in a hypotonic, hypertonic, and isotonic solution. So take a look at this video and see if your predictions were right in lab this week. This talks about how things are happening in a plant cell, okay? We've already looked at this once. So water's flowing out, the cell membrane is going to pull away from the cell wall. If it is in a hypertonic solution, if it's in a isotonic solution, we have an equal movement of water in and an equal movement of water out. And that plant cell is going to be flaccid because we're not putting pressure on the cell wall. And the vacuole, the central vacuole might not be 100% full. And then in a hypotonic solution, water is flowing into the cell. That central vacuole is full, and the cell membrane and the cytoplasm and everything is putting pressure on that cell wall, which is making that cell turgid. And that is where we generally like to be with a plant. That's where it's standing up nice and straight and tall, okay, and hanging out, okay? Plasmolysis and Eladia. And now this is your turn to practice. I would take time to put this on a piece of paper, okay, and figure out what is going to happen, okay, with this cell when it's going to be placed into placed in that solution. Which way is the water moving? Okay, so it's water that's moving because that's a selectively permeable membrane that doesn't allow that sucrose or glucose to cross, okay, the membrane itself. So this is going to be movement of water. So figure out which way that's going to move, and then if you have any questions, come and talk to me about it. All right, so that finishes up just straight, simple, passive transport. In the next video, it's going to have facilitated diffusion.